Welcome back to Muscle Serpents University. I'm Dave Palumbo, and today we have a very special guest. We're going to be talking carpet pythons, genetics, and God knows what else. We maybe even bodybuilding a little bit. He really needs no introduction. He's the author of The Complete Carpet Python. I want to welcome Nick Mutton to the show.
Good to be here.
Nick, is this your TV debut?
What?
Is this your TV debut? I mean, you usually do radio shows and podcasts.
It is. I've done countless podcasts, but this is the first time with video. So,
are you? Is this your house you're in? Or are you actually in your snake building, or is that the one in the same?
Now I'm in my office downstairs.
Now, Nick,
previously working on some academic papers and filing some pictures for the new book.
You're arguably the biggest, breeder of, I guess you could say, Australian pythons in the world, I would say. Would that be accurate?
I mean, it would depend on how you define that. There's people that might produce more of this or that, but I would say I probably have the most genetically diverse collection of that kind of stuff in the world.
I hate to ask this question because people ask me this question. I say I don't know, but how many snakes do you think you got at this point?
Who even knows?
Yeah.
I don't know. It changes every day. It'll be, yeah. I had to ship some today. It'll be a few less, thankfully, today. But this time of year, probably about 1,000 with all the babies.
Now, what year did you write The Complete Carpet Python?
It took 2 years to write. I think we published that in the middle of 2011.
Wow, yeah, and I read that book. It was one of the first books I read right after getting into carpet pythons, and it was an overwhelming amount of information, but it was one of those books that I like to call like a toilet bowl book. You sit on the toilet and you read a couple pages every time, and you just keep learning more and more and picking up things you didn't catch the first time.
Well, the first edition sold out completely, so Justin and I are working on an expanded second edition, and I asked Bob. I was expecting maybe him to be more resistant to make it larger, and turns out he's totally on board. So now the first edition was 360 pages. The second edition we're shooting for 500 pages. And it will be as in-depth as you thought the first edition was. It will be exponentially more hardcore than that. Warren's writing an entire molecular phylogenetics chapter section of it. So there'll be proprietary genetic testing done just for this book.
Warren Booth.
Which is... I wanted to do something that no one has ever gone that far for a reptile book.
Yeah.
To actually have, sequencing the full mitochondrial genomes of all the carpets.
Unbelievable.
It'll be that level.
You sequence the mitochondrial DNA, which comes from the mother, essentially. The mother, you always inherit the mother's mitochondria, which is the organelle inside the, all your cells that controls, energy and fatty acid oxidation. But that has its own genome, and you actually are sequencing those, correct?
The entire thing.
Why is that, though? Why would you go for the mitochondrial versus the regular nuclear,
Oh, we're doing
genome?
both.
Okay.
The mitochondrial genome is a lot smaller than the nuclear genome. Sequence the entire nuclear genome would be pretty unfeasible at this point. We'll be sequencing quite a number of nuclear genes and all of the mitochondrial genes. So it'll be a depth of sampling at the genetic level that no one's ever done on carpets. No one's gone that far.
To see how related these different species are, or not related.
Yeah, it turns out like cladistics, the kind of organization of different species and how they relate to one another, is considerably more complicated than was previously believed. We're just now starting to realize that what you have it's referred to as genetic admixture, basically hybridization events between distinct forms and species. That plays a much bigger role historically than we really realized. Things diverge from each other and speciate at different times, but then might come back together at other times and exchange genes again, and then be separated again. That sort of thing happens a lot, and certainly has happened with carpet pythons, making it very muddled and difficult to tease apart the relationships between forms. Even with our own species, we now know that we, as Homo sapiens, interbred with at least 3 other non-Homo sapien species.
Right. Neanderthal
at least
being one.
In recent times. There's a little bit of a percent or two of a lot of other species of hominid in us still from rather recent interbreeding. So and snakes are certainly no different.
Let me ask you this question, because you're a purist. You like to really keep your localities and your species separate. You know, you don't like the crossover. You don't like the, well, as we say, hybridize. But now that the classifications have changed, are your whole thoughts about that changing? Because, you know, certain species are turning out that they're not separate species, that they're the same species. Other species are not this part of that species. How does that change your outlook on your breeding projects?
It doesn't, because mine are broken down at such a finite level that literally nothing would change in what I pair.
Oh, really?
I will only, as far as the only reason people hybridize carpet pythons or boa constrictors or anything anymore is purely to get morph A into morph B. And I'll go as far as I can go within one well-defined form. I won't, with carpet pythons, I'll happily make an xanthic hypo coastal carpet because there's not really any doubt that they're both coastal carpets. I would not breed where it gets murkier and where I'm conflicted in that when this book comes out, what it's looking like based on the preliminary results, and it's likely to just continue on that trend, is that several of the northern forms that are previously recognized as separate distinct forms are actually all the same species. But they're not, but that's not the end of it. It's not, if a Darwin carpet, a New Guinea carpet, and a carpet on the Cape York Peninsula are all genetically close enough to be considered one species, it doesn't mean they're the same snake. They are distinct regional populations. At the genetic level, you can tell them apart genetically. They all cluster with their own type, but they're not, that level of divergence is not deep enough to necessarily warrant separate taxonomic status.
Gotcha. It's like boa imperador. It occurs all over the place, but yet there's different regional, you know, localities of them. Yeah, these three distinct populations within what is likely an expanded variegata are all very distinct, but it's probably not quite distinct enough. And that would mean that on a technical level, if they're all variegata, you could make an albino granite would be pure. A pure variegata. It would still be a mixed blood snake because you have to, these populations are not, you know, and the two most distantly related are the New Guinea animals and the Cape York animals are more closely related than the Darwin animals. Interesting. And if you look at the historical Pliocene and Pleistocene ice ages and interglacial periods and its effect on sea level, and you can see when these areas had broad contact and gene flow and when that was gone, you can see it's probably that stuff in the west around Darwin was separated much earlier than the stuff closer to the Cape York Peninsula, which maintained gene flow presumably in contact up till about 8,000 years ago. So basically what you're saying is Australia and New Guinea at one point was one landmass, and then when the oceans rose, obviously they got separated and the snakes were separated. It's even more than that. Southern New Guinea is Australia. Everything south of the Owen Stanley mountain range, which cuts New Guinea north-south, everything south of that is Australia. Really? Geologically, it's called the Papuan Basin. It is it's part of the Australian tectonic plate that when it collided with the Pacific plate, it pushed up those mountains and basically the seafloor buckled and it lifted that submerged part out of the water to form Southern New Guinea. But geologically, it's Australia. And for most of the last 6 or 7 million years, it was all dry land. Right. It's a minority of the time that it has been submerged. And that's why you have, you know, spotted pythons and carpet pythons and kangaroos and all these typically in blue-tongued skinks in that southern bit. If you go there, it looks like Kakadu National Park, but it's in New Guinea. It's a dry savanna, floodplain environment. It's like a little bit of Australia because it actually is Australia in a geologic sense. So. Now, locality is very interesting, and obviously there's so much to still be done. But you know, let's face it, we're a morph crazy, you know, hobby now. I mean, everyone wants to see what the latest morph is. And you know, my belief, you know, I wanted to just get your philosophical belief on something. Do you believe that we live in an intelligent universe, or do you think we live in like a random universe that just things happen because the atoms collide in the void? I think all evidence suggests the latter, not the former. That much of life and success, and particularly in evolutionary terms, is largely luck. Did the situation changes, and if a beneficial mutation that helps an organism cope with that change occurs within enough time, they adapt. If it does not, they become extinct, and it's kind of a random roll of the dice. There's been a lot of really successful animals that went extinct
the key to extinction seems to, and survival seems to be being generalists versus specialists. If you animals that become too specialized, can be a very good way to make a living for a short time, but as soon as that dynamic changes in any way, they can't cope with it fast enough, and they tend to become extinct. Whereas generalists are more adaptable. It's why you don't see, in North America, we don't have any short-faced bears, the largest bear that ever lived.
Yeah.
We don't have those anymore. We don't have a lot of things anymore because the things that they ate became extinct. They couldn't eat smaller prey. They went away when all of their prey went away.
But when we domesticate animals, Nick, it seems like all of a sudden this huge variety of color palettes become available that we almost can't fathom where they can. Look at ball pythons and reticulated pythons. Look at the betta fish, the Siamese fighting fish. I look at some of these colors, compared to when I was a kid when they had, blue and red. I mean, it's almost unfathomable that, where did this come from? It's like we're almost like our will to want to see these morphs have actually made them come into being almost.
In large part, that is true. There's a little more to it at the biological level. The longest running scientific experiment in the world was started in the Soviet Union, and it's still continuing to this day, like 60 or 70 years. And they wanted to study how humans domesticated wolves and made wolves into dogs.
Right.
So they started with a completely wild canid, a fox, which they were already breeding for the fur industry even back in the Soviet days. Mm-hmm. So they started domesticating these foxes, and what they found is that certain characteristics we associate with dogs are the result of adrenal gland output. Wild animals have a higher adrenal gland output. It increases their fight or flight response and their survival odds. Domesticated animals, when we domesticate a species, one thing we are, whether we consciously do it or we unconsciously do it, we are selecting for mellow, tame, workable animals.
Right.
Those animals invariably have lower adrenal gland output, and what the Soviet and Russian experiment showed was that when adrenal gland output falls below a certain point, color and pattern mutations start popping out.
Really? Interesting.
In particularly piebald, there's a strong correlation between piebaldism and lower adrenal gland output.
Wow.
Now, if you think of any domesticated animal you can even imagine, they always have white patches. They are all piebald. Think of every dog you've ever seen in your entire life. I couldn't think of a dog that didn't have at least some white patch.
Yeah.
All of that is piebaldism, and they're the same with their floppy ears and their tails that curl over. All of that comes about at lower adrenal gland output in canids.
Interesting.
So these mutations popping out in captivity anyway, it's partly because we are domesticating them. And in ball pythons, they're essentially completely domesticated. Another line of evidence for that is that one of the other hallmarks of domestication is easier and more frequent reproductive events.
Yeah.
Wolves only produce 1 litter of pups a year. Dogs go into heat twice a year. Mm-hmm. I have seen ball python. I have a friend that bred a male ball python that was literally 3 months old. 3 months. And it weighed 250 grams. I think 247 grams at sire to clutch. That is not natural. Right. To be 3 months after you leave the egg producing offspring. That's clearly a domesticated.
Right.
It used to be, I remember in ball python, I remember when they were hard to breed.
I remember when they were hard to keep alive.
Yeah. Now we've just engineered them to be better captives. Now they require no temperature cycling. They're effectively non-seasonal breeders, and they'll breed at ridiculously small and young ages with no external stimulus. But 20 years ago, that was not the case.
Yeah.
Some of the carpet python lineages are getting to that point. Boa constrictor, some of these long-running species people have been working with for a long time are starting to get there, where sometimes I get younger guys in the hobby now, like trying to tell me how it is. And they're like, oh, you don't need to cycle your snakes. You don't need to do all that. It's like, but these dinglings don't really realize that the goalpost has been moved. The reason why it's easier now is because of all the guys 10, 15, 20, 30 years ago.
Sure.
Every generation of captive breeding further down that track you get, those goalposts get a little, you just don't need to, they move, and it just gets a little bit easier until eventually they will just be non-seasonal breeders as well. I'll just do it whenever.
Right. I still think it's easier to have a breeding season just because if you have random things breeding, you can't keep track of it all. It's way easier to keep them in I find at least to have your whole collection cycle together.
With the Australian stuff, I can firmly say unequivocally that if you cycle them, you will get more consistent, predictable results.
Yeah.
You will get usually better fertility, better fecundity, and a more predictable outcome. If you try not to, it's like anything else, and you know this from your, past career as a bodybuilder. You get out of anything what you put into it. Sure. If you put in a half-ass effort, you will in fact get a half-ass result. And if you're happy with a half-ass result, then proceed, I guess. If you would like to have good results, consistent results all the time, year in and year out, then it requires a little more effort.
Yeah, and everything likes to be done in a cyclical nature. I mean, so the snakes obviously respond better. I mean, it's no coincidence that if you breed a snake, and I have a female, ball python or carpet, it's almost like they lay a clutch the same day every single year. It's unbelievable how regular they are.
I had an Australian olive python when I built my snake building, because all my snakes were in my house many years ago, and then I built my snake building out back, and the building, because it was built by a contractor, so of course that didn't go well, because those things never do.
No, never.
With contractors, the usual story: take half the money, disappear for 6 months.
Yeah, yeah. They do 90% of the job, and they just don't want to finish the rest of it. Yeah.
Yeah, and then spend your money buying supplies for some other job or something. But so the building didn't get finished until 3 months later than scheduled. So I didn't start cycling until 3 months after the time I normally did.
Yeah.
It resulted in the absolute worst season I've ever had. I produced 82 animals out of giant, literally, because that window, those animals had been cycled at the same time. They were in that annual rhythm of follicular development, and it was way past. I did get a clutch of Australian olive pythons, and by happenstance, I grabbed I only have a handful of really large egg boxes, and they got panes of glass on the top of them. I happened to grab the exact same one that her clutch from the year before that snake laid on the exact same day.
Wow.
Exactly the same day, a year later, even though I moved the cycling 3 months, it laid on the same day. So that train had left the station long before I did anything.
Right. Well, and olives tend to be a little later, right? They tend to be like spring breeders?
They'll breed at the end of the winter.
Yeah.
Some of the stuff, the more cold-tolerant species tend to, like inland carpets, Brisbane coastals, Brettles pythons tend to breed more into the spring when they warm up, because their winter temperatures in the wild are so cool that they are not terribly active.
What is the secret with olives? That's one species I've really just had problems getting them to go. I don't know why. It seems like I'm missing something.
I'm going to give everybody the secret right now, and then everyone will start breeding their olive pythons. They're getting them too big and fat. It's the same as same problems people have with their boa constrictors. They get them too big and fat. They might get something. The snakes are dead when they're 10 years old or younger, because they're not supposed to be that big. An 8-foot olive python should look like, basically, imagine an 8-foot California king snake. That's what it's supposed to look like.
Wow.
It's not supposed to be, when they get too fat, they don't ovulate, and it just never. And people, part of this I blame is the kind of the morph craze of the last 20 years, and that people equate larger size with more eggs and more profits as it pertains to morphs, and that applies to everything. And a lot of times with some species, you're just going to kill them. They're not.
Sure.
You think these olive pythons are $1,000 a pair. Well, this giant female will lay more eggs than this more modestly sized one, and they err on the side of size, and they end up getting nothing. And animals that end up living a substandard and shortened life expectancy.
Do you think that increases the I've noticed that I saw there was a post on, I think it was on Facebook or Instagram. People were talking. Someone was talking about that that they found that I think it was Casey had mentioned it, that his Brettles pythons had tumors or something like that. Had a tumor, and he was saying. And then other people chiming in. Yeah, my carpet pythons had more tumors. I noticed I had 1 or 2 snakes that died of tumors as well. Does any of that have to do with morphs, or is it more related to the feeding over response that you were talking about?
I don't even think it's that. I think that if you keep a snake alive long enough, they get cancer.
Yeah.
Just like what's the number one killer of people when we get old? Cancers.
Yeah.
Of various types. I think the problem is I don't think it's more common in carpets than it is in any other group. I think that I notice it a lot more in carpets because I just have so many carpets.
Yeah.
The number one killer of snakes in my collection is cancer. I have not had a snake die of a respiratory infection in probably 15 years, but I lose 1 or 2 to cancer every year. But I have hundreds of adults.
Right.
So that's expected, and it's usually an intestinal tumor or something along those lines.
Yeah, it's like a blockage, and then they can't eat, and they wind up regurgitating. That's what happened to one of mine.
I've seen it in a ball python. I've seen it in a couple of green tree pythons. I've seen it in other things, but my sample size is so skewed towards carpet pythons that, of course, that's where I see it more. I have rough scale python. My breeder male rough scale python just died. It had a giant malignant tumor. So it happens, but usually it's in older snakes. I mean, Casey's Brettles python, that snake was, 11 or 12 years old, which is it's not necessarily old for something that can easily live 25 years,
right.
But it wasn't, a young animal.
Sure.
I think a lot of people have come to expect, and part of this goes back to the morph thing too. They make these things breed as young as they can, as small as they can, and they breed the crap out of them, and then they die, and they start to think that that's normal. Most people watching this video probably think that pythons live 8 or 10 years.
Right, right.
When they should live 30, 40, maybe even 50 years.
Wow.
But they don't do that if you're pumping them up to breeding size in 18 months and breeding them every year for 5 years. You're just burning them up.
Sure, sure.
So most keepers probably don't keep an animal alive long enough or in their collection long enough to see these things that happen later in life.
That's true. That's true. So getting back to olive pythons, you think the trick is not getting them as fat? What if you have an animal that might be? Can you put them on a diet? Like I always cycle feed my pythons. Like I don't feed them for 3 or 4 months out of the year while I'm trying to breed them, and then I'll give them more food on the off time. Is there a magic in terms of temperature to get olives down to that is different than carpet pythons, say?
No, they're in the same building as everything else, right next to this carpet stacked on top of them. Like they're all around.
Really?
I treat them the same. They're in bigger cages, of course, but they don't need to be as big as people think, I guess.
How often do you feed yours?
Oh, well, the males I hardly ever feed.
Right.
It generally sucks to be a male of anything at my house. Well, honestly, that's another thing. Like big fat males are lazy breeders. They don't breed well. They don't live very long. There's no advantage. The males that get the job done are the smaller, lean and muscular, but small. They live forever. They don't eat much. They don't crap much, and they breed like crazy. It's true. Like all my males are males are actually the larger sex in all the pythons, but my males would never know that.
Right.
All my male olive python breeders are, maybe 6 feet long if you really stretch them.
Wow. Yeah. That's
the diameter of a 50-cent piece, and that's it.
Wow. That's small. Yeah.
And some of them one of my males is 20 years old.
But don't you find that the females can eat the males if they're too much smaller? If the size discrepancy is too big?
Oh yeah, they totally could, absolutely. But I've never seen that been a problem in all these years of working with olive pythons. Even among more cannibalistic species like Papuan pythons, which are well known to eat each other, it seems to be a mate selection criteria. Females will either accept a male and let him breed regardless of his size, or they just don't like the way he smells, in which case they'll try to kill him. And it doesn't matter. Like a lot of times, like you put in a similarly sized male in there and they'll attack it, but you put a little tiny male and they'll let him do his thing.
Right. I
don't know what criteria they're using, but they're clearly doing something. I had ringed pythons are horrible for that.
Yeah.
I had a trio for years, and the females would never let that male. They would immediately try to eat him every time for 5 years. I finally just had to separate the group to different people.
Yeah.
They were never going to accept that male, and he's only alive because I had to stand there with a bucket of water and ended up putting him in there. And it just other people that get a pair that they like, they'll keep breeding for you. Do you
winter your olive pythons together, or do you put them together after you've cooled them?
I put them together while I'm cooling them. I cool them for about 6 weeks, and when I'm about in the middle of it, I'll start putting them together.
Yeah.
A really, really, really effective thing, one of the most effective things to get olive pythons breed is male combat.
Yeah.
The problem with male combat is if you combat like carpet pythons, they go through this very ritualized wrestling match where they try to wrap around and knock the other guy down. It's very ritualized. One eventually turns away and leaves. Olives, it's a little bit different. Basically, you put them in the cage together, and you'll know the very second that one male realizes there's another male in his cage, because it'll crawl across his back, and he'll arch his back up like he's trying to throw him off, and then you separate him immediately. Because at that point, you're 10 seconds away from bite, constrict, and kill. It's not a ritual thing. They will kill each other. Like,
really?
Every time they will kill each other.
Interesting.
I have friends that have had males kill each other. They will 100%, they'll kill each other.
Interesting. So you don't want to leave them alone unsupervised.
No. You pretty much got to stand there the whole time, and once you can see them starting to get agitated and recognize the presence of the other male, immediately separate. Don't even let it get very far.
So it's enough just to get that little bit of behavior together.
Yeah, and it's basically the basic premise was the same with carpets or anything else. So it's like, well, even if this male didn't want to do it before, he's certainly not going to let this other guy do it.
Right.
So it definitely lights the fire under him to get the job done.
Let's talk genetics now, because I find this topic fascinating, and I've heard you talk about it on other podcasts, but I think it's worth going into, just because I don't think people really understand what morphs are. I think they think they're some kind of magic snakes or something like that. They don't realize that they're basically defective snakes in a sense. There's something defectively going on with these snakes, and you kind of really simplified, made it simple and broke them down into color morphs versus pattern mutations. Give us that explanation, because I think it's worth repeating.
Okay. Basically, all morphs are broken genes that don't work right. But what a lot of people don't realize is that most genes play a role in multiple different, seemingly unrelated jobs. So while we focus on the color and or pattern component of a gene product, it might also be involved in other things that don't seem to even necessarily make sense to us. And this is where you have genes that have problems. The scoria boas, jag carpets, spider balls, this kind of stuff, super zebra jungle carpets with their tail issues. These other, why would a gene that makes your heterozygous form makes your pattern really busy pattern, your homozygous form makes you patternless and messes up the tip of your tail? That gene is simultaneously involved in pigment distribution and the formation of tail vertebrae, of caudal vertebrae.
Right.
Why are these seemingly unrelated things? But that's when you have morphs that have baggage. It's because the basic idea is this: if the gene is not doing its color and pattern job correctly, it's not doing any of its jobs correctly.
Right.
And if some of those more ancillary functions, are not working right, that is where you sometimes get into trouble on occasion with some morphs. Mm-hmm. As far as the broad categories of mutations, you could divide what we see in herpetoculture into two different groups, and those are pigment synthesis mutations and pigment distribution mutations. Pigment synthesis being the ability or inability to properly produce pigment, but the process, that is an entirely different process than the distribution of that pigment.
So the pigment production would be your T-positive albinos, your T-negative albinos, your axanthics, and your aneries?
Yeah, exactly. All of those sorts of mutations. And then the pigment distribution mutations are where you get into trouble, because when you rearrange this is going to get complicated. Basically, you have to understand how the process of pigmentation works during embryogenesis. Pigment cells, or pigment precursor cells, are called melanocytes.
Right.
Melanocytes are processed in the neural tube, and then they migrate out of the end of the neural tube to various predetermined points along the embryo. They set up shop, they divide, they proliferate, and the animal becomes pigmented. For whatever reason, they cannot migrate by themselves, so melanocytes co-migrate. They basically hitch a ride with other cell types that are migrating to different points in the embryo, and that's how they get to their destination.
Nick, I wanted to stop you because in case people are not following, embryologically, when an organism is developing as an embryo and the cells are growing and the spinal cord is growing, or the primitive spinal cord, and all the cells are going to where they have to produce the arms and the legs and all the different stuff. What you're saying is the pigment cells kind of piggyback with the other cells that are moving, so that the pigment gets distributed according to how the other aspects of the animal develop, spinal cord and brain and stuff like that.
Absolutely.
Okay.
And so when you rearrange where the pigment goes, you are, at some level, rearranging whatever it was hitching a ride with. Yeah. And that can get you into trouble. If you look at all of the morphs we see in herpetoculture, regardless of species, doesn't matter if it's a fat-tailed gecko or a carpet or a boa or whatever, all of the time, every time you have a morph that has problems and has some baggage associated with it, it is always the distribution mutations.
Right.
It is very, very, very rare. There are a few examples where you have a pigment synthesis mutation that has its problems, like the original caramel albino ball pythons and borderline sterility and spinal kinking, but that's pretty unusual. It's almost always the pattern mutations, the pigment distribution mutations, because of that.
And you know what? I think the original caramel albinos were just inbred, to be honest with you, because I have a line of caramel albino ball pythons that never ever have any problems with them. They breed, they're always healthy, they look great, and I got mine from Vin Russo originally, most of the ones I have, and he also told me his never have a problem. I think there was, you know, he outbreeds a lot of stuff. I think there was a lot of inbreeding in the initial caramel albinos.
Well, that's a weird topic, and I happen to have weird insight into those particular exact animals.
Oh, really?
Early on, there are two different types of caramel albino that are genetically incompatible. One type has problems, one type does not have problems.
Interesting.
And way, way back, no one knew they were different. They knew they came in separately. They were bred together one time and effectively produced double hets. Those double hets got scattered into the ether, never to be seen again, but they didn't die. Ultimately, yeah. Vin ended up with some of those. So he had both types of caramel albino in the same animals. He was producing both types independently and even animals that were both types simultaneously, which were visually distinct. Yeah. So the easier explanation is that you have the good type. That doesn't. The other type doesn't have any problems at all. Yeah. It's amazing, eggs, right? Females breed. They don't have kinks. They're perfect. Yep. They're very similar. That type I tend to think is better looking. Yeah. As well. So, yeah, they're. But that's kind of a. With that one possible exception, it's always the pattern mutations that distribute pigment.
Nick, is it possible that the mutation is not of the pigment distribution, that the mutation is actually in the neural development, and that the pigment distribution is just a side effect of that?
Oh, it could be. There's really no good way to answer a lot of these questions definitively. All you can really do is look into other things that have been studied and draw inference from that.
Because could it be like the JAG gene is really a neural defect, and it just so happens that the animal looks super cool, you know, because of that?
Instead of rearranging the pigment and also rewiring some brain neurons, it's actually buggering up the brain neurons, and that's affecting the pigment? Yeah. It gets you to the same place, but that's completely possible. But it's
like we're breeding, like, you know, neurologically challenged animals, basically, because they look cooler, you know?
Well, that is with the jaguars, I did a lot of research doing the book the first time and trying to figure out why does this happen. And there are other examples of similar mutations in mammals that have been studied in a laboratory setting, and what you find is that for whatever reason, there is those melanocytes, those pigment precursors, attached to certain types of brain neurons, and so you're really rewiring their brain a little bit with some of that stuff, and that is the likely cause of this. A good analogy you can look at for that sort of thing is Dalmatians, dogs. For the longest time, the AKC, and still, like, the breed standard is to not have any black spots on the ears. Mm-hmm. Dalmatians are also famous for being deaf. Turns out that there's a strong correlation between the amount of black pigment on the ears and the incidence of deafness. The more white, the more incidence of deafness. Wow. So they were inadvertently, because they didn't understand anything, encouraging and advocating for deaf dogs. Oh, shit. Yeah, I didn't know that. Because for whatever reason, there's pigment precursors inside the inner ear that, like, I don't know what they're doing there. Right. But if they don't make it there, then the ear doesn't work right. Gotcha. So it's one of those sorts of things.
Why the piebalds seem to be pretty normal in all the species, and that's a big pigment distribution, you know, screw up kind of?
I think it's just that actually the piebald mutations really illustrate how that whole process of pigment cell migration from the neural crest and embryogenesis, how that plays out. The neural tube, which is where all these cell types are made, roughly corresponds to what will become the spinal cord. Right. And the opening is basically at the base of the head. So when these cell types leave, the first place they get to is the head. The last place they get to is the belly, and the back is kind of dot dash all the way down. So when you interrupt that process, that's why a piebald ball always has a pigmented head and never has a pigmented belly, because it's basically, it's like imagine a light switch on a wall where it's supposed to be, and that light switch represents the process of pigmentation. Mm-hmm. You flip that switch, is supposed to be turned on at a certain point, pigment starts going out. Eventually, the animal's fully pigmented and the switch is turned off. What a piebald mutation does is it's basically the middle ground between a leucistic mutation and a solid black mutation. Basically, the leucistic mutations, it just shuts the switch off almost immediately. Basically, no pigment goes out, white snake. The melanistic mutations, the switch is left on too long and pigment keeps going out.
Right.
And it overpigments. And a piebald mutation basically shuts that switch off at some random point in the middle. Since the belly is the last thing to get pigmented, the belly will always be white, and the head will always be pigmented because it's the first thing. And that's why you end up with the dots down the back, a white belly, and a pigmented head. If you look at the same exact form of piebald occurs in our feeder rats. Most of the rats we feed off, we're feeding piebald rats to piebald snakes. Think of a hooded rat. What is it? It has a white belly, a black head, and intermittent amounts of black on the back.
True.
Identical in its genetic makeup to a piebald ball python.
Right.
That we're feeding it to.
Yeah.
Think of a Holstein dairy cow. Same thing. White belly, black spots, black pigmented head.
Take that new piebald blue-tongued skink they just found. We have a picture of it we could throw up there.
Yeah.
Black head, white belly.
Yep. Because
with the stripe down the back.
The bottom is the last part to get pigmented, and the head is the first part. So it always have a fully pigmented head and no pigmented, not much pigment on the belly.
What do you think about that piebald blue tongue? Have you taken a look at it?
I have. I had like 20 people send me pictures of it to make sure I saw it, but I gotta say, like, I don't care, honestly. Like, it's as an Australian skink, they're gonna do with that what happens with everything, where they're gonna hybridize that thing into oblivion by breeding it to every other skink morph in every other species with no regard to establishing it as in the species it was originated in.
Right.
And at that point, to me, it's like I've lost all interest, and unfortunately that's played out. It's played out in boa constrictors. It's played out in a bunch of things. What's
your prediction? How long will it take to get to the U.S. that thing, the offspring?
I don't know. It does seem that eventually things show up one way or another. It's not always like, a lot of times the story is not as sexy as people think either. It's it's sometimes pretty mundane, really, I mean, a lot of the stuff, like I've got rough scale pythons and albino olives.
Right.
Those legally left Australia from a privately owned zoological facility and went to a privately owned zoological facility in Sweden that then sold them legally in Sweden, which is legal to do under Swedish law. So no laws were broken at all.
Right.
It's an unlikely story, but I've got the documents prove this. This is what happened. It's not. Sure. Sometimes a surprising number of things enter the pet trade in the U.S. and Europe through confiscation. Typically what happens if somebody tries to smuggle something into Europe, which does happen from time to time, and very often, most of the time, they get busted at the airport with a carry-on bag full of whatever. They rarely euthanize those animals.
Right.
They typically, most European Union countries will look on their books and see who they have that's a licensed breeder of that species. Australia won't take them back for quarantine reasons, and the German government, say, doesn't have any interest in keeping some skinks.
Sure.
They loan them to a licensed breeder of that thing, and those animals are basically more or less property of that country's government. They're on permanent, indefinite loan, never to be returned. And if they were to accidentally reproduce, then those animals are legal to be traded in the pet trade. And wouldn't you know it, they always accidentally reproduce. A German breeder, nice enough guy named Mark Mensa, has been breeding rough-scale pythons and albino Darwins for years because somebody got their carry-on bag searched at the Munich airport, thought they were going to go to the Ham Expo and make some quick money. The German government looked up who they had. Mark wrote a German language carpet python book, gave them to him, and he's been breeding them and selling them legally ever since.
That's crazy.
It's not always, a bunch of cloak and dagger stuff.
But isn't it also wild? I mean, you have, you have like some amazing Brettles pythons. I got a pair from you. You got the stonewashed with the stripe, genetic stripe, with the hypo. I mean, they don't have that in Australia. It's crazy. I mean, we've produced morphs out of Australia when this species originally came from Australia, and we have better stuff in America. It's wild to think about the fact that things can mutate in a person's collections, and we could actually have better stuff in the country, out of the country that the species originated from.
A lot of mutations are not. With carpets, there's very little wild-caught carpets. Even in Australia, it's mostly people breeding legal captive-bred animals.
Yeah.
stuff pops out. I can't really talk about it or show pictures of it, but I got corresponded with a guy who's sending me some pictures of some wild carpets for the book. And he just hatched out the craziest, most insane, he hatched out my dream snake. And I'm helping him kind of figure out how it works genetically, because there's more layers to it. But
what's your dream snake?
I can't tell you.
Oh, give us a little teaser, man.
It's very dark.
Oh, melanistic, really? Wow.
But in this instance, there were a few of them in the clutch. But it also looks like it is likely a parthenogenically produced clutch.
Uh-oh.
Which adds a layer of difficulty and complexity along with that. And so I'm helping him figure out and decipher, because they don't have a lot of stuff like that down there, but we have, we know how those sorts of mutations work here because we've got them in numerous other species.
But the boa's got the great IMG, morph, that makes a black boa.
But when you look at those, oh, I know, that's my dream boa. I remember seeing the very first one. There's a guy walking around at the NARBC show in Arlington, Texas, a very long time ago, and he had this. He was just like, "What do you think of this?" And he didn't seem to know. It's like, I think it's the most amazing thing I've ever seen. You take it home and quit exposing it to God only knows what's floating around at this reptile expo.
Exactly.
It was like I was just dumbfounded at how amazing it was, and that he was like walking around seemingly confused as to what he even had. And it was like, I mean, that is a cool snake.
Yeah.
So, but that kind of stuff, those sorts of mutations are always the homozygous expression of that gene. They're never the heterozygote. They're always the homozygote.
Interesting.
So you never see a solid black het for something. That's always the visual, the homozygous, and they're almost always incomplete dominant. They basically are always. It can be a subtle incomplete dominant where it might be pretty hard to tell, but there's usually indicators.
But in the boas, you can have the single copy of the IMG gene, and you will get a black snake, more or less. Whereas I know, like with the blue tongues, the melanistic blue tongues, it seems like you need 2 copies to get that.
Yeah, there's multiple incompatible types of black snake mutations in ball pythons that are not allelic, and they work under slightly different mechanisms.
Yeah.
In those cases, they're all incomplete dominant, and your heterozygous form is darker, but it's certainly not solid black or patternless. I mean, there's always variations of these things, but the basic outline of how do you make a melanistic animal, it's going to be an incomplete dominant of some sort.
Yeah.
The one that's screwy that people have a hard time with is hypomelanism, because hypomelanism can be either, because there's 2 entirely different types of hypomelanism that have nothing to do with each other.
Explain it.
They're very different. You have a pigment synthesis type and you have a pigment distribution type. But the definition for hypomelanism literally just means less black.
Right.
You can get there 2 different ways. It has become fashionable lately to call the pigment synthesis type T-positive albino, because that just sounds sexier.
Yeah.
But the difference between a hypo and a T-positive albino is literally nothing. It's the same thing. But T-positive albino sounds way cooler, so we tend to use that term.
What's in carpets, what would you call the hypomelanistic versus the caramel albino?
Okay, in carpet pythons, you've got both types now. You could probably lump the caramel mutation in coastal carpets under the kind of pigment distribution hypo umbrella. They're not really hypo-y, but they're along those same lines. The true hypos, which I've got, but those are definitely incomplete dominant pigment distribution. They can make black, and the black that they will eventually gain can be jet black. It just affects the quantity of the black, not the quality of the black. A friend of mine, Troy Bromey in Australia, just produced and proved out. I've been working with him for like a year and a half behind the scenes and everything, kind of helped coach him through proving this thing out. The other type, it's a pure Darwin, and he is a pure Darwin hypo that is the pigment synthesis type, the quote-unquote T-positive type.
Right.
It's completely different. In those instances, the black is basically like a purpley color.
Wow, that's awesome.
The surface area of black is unchanged, but the color of that black is diluted. They're amazing looking, but but he's got people who don't understand this, like endlessly arguing with him on the interwebs about that, implying that his hypo Darwins are just the hypo coastals bred into a Darwin. It's like, no, it's a completely different type of mutation that is just equally under the same name of hypomelanism, but it's completely different. And you see that in boa constrictors. You've got multiple T-positive lines. Yeah. And you've got the traditional hypo boa is the incomplete dominant pigment distribution type.
Right. So which are would you say the hypos, the true hypos in the boas would be the ones that would have the problems, you know, because it's a pigment distribution mutation?
I mean, if they were to have problems, they would be the ones more likely to. But I think, talking to my boa friends, they all seem to be fine, though. Just because they could have a problem doesn't mean they necessarily will.
I think they're fine until you combine them with like something like a scoria or something that, well, I think then it can make it a little worse.
Sometimes you get people talking about, like, and I'm sure you probably heard it from boa people, that if they just add some other morph to their scoria, it'll fix it. Which is just, you know, madness on its face to even say that. But it's like, look, you have a broken snake. Adding another broken gene on top of it is not going to make the first gene fixed.
It makes it worse.
It's going to, it could make, you know, it's probably going to do nothing, but you could see a scenario where adding another broken gene might make something worse.
It will make it worse.
It will not make it better. And there's
absolutely
ample evidence of that in carpet pythons. And I hate to be the one to tell people this stuff, but it's, you know, the jaguar gene is the same thing.
Yeah.
As a scoria boa. If you make an exanthic jag, it will aggravate the neurological issues. It'll make it more likely to present that sort of symptoms, and the severity will be greatened. If you make an albino jag, if you make a granite jag, it makes them worse.
Yeah, I agree with you. I agree. 100%.
It does. I've never seen a granite jag that wasn't kind of gurked out. Like, I've had one exanthic jag that wasn't too bad, but a lot of them, you see an earlier onset of those sorts of symptoms, and it doesn't help.
Yeah.
Other mutations like, you know, caramel and hypo, which don't have any effect on it. It's still the same. It's the odds and severity of symptoms as far as the neurological stuff are unchanged. It doesn't make it better, but it doesn't make it worse. But some definitely seem to make it worse. Yeah.
It's amazing, though, that these animals, whether it be the spider ball python, the carpet, you know, the carpet jag, or the scoria boa, they all eat and breed amazing. It's almost like they're better eaters and better breeders. I don't know why, but you know, but they seem to.
I've never had any. None of them have any other than the. Yeah, I get conflicted. I wish they didn't have the wobbles and this kind of stuff. I wish that was not the case.
Yeah, me too. But we all.
I'm conflicted about producing them, but at the same time, it is not painful to them. They seem completely unaware that they're even doing it.
Yeah.
They don't understand that what's even happening. So it doesn't seem to have any effect on reproductive success, longevity, or quality of life. You just wish you didn't have to see it.
Yeah.
So
I know sometimes when they get older.
I would highly discourage anyone from trying to make an anery scoria. I'll just tell you, it's probably going to be a train wreck.
Yeah.
Don't do that. Like, that's probably not going to go well. But if you use other precedent in other species, a hyposcoria probably wouldn't have any effect.
Yeah, I have a hyposcoria, you know, the mother, I have a regular scoria that has no problems whatsoever. I don't see any shake to it really much at all. I have one that's a hyposcoria and jungle. I don't know if it's the jungle that did it. It's a little bit more wobbly, I notice, than the regular.
My gut would say the jungle is probably...
Yeah, the jungle probably is the thing screwing it up. Yeah, I would agree.
You know, just because something could be doesn't mean that it is, but it is... I think it's foolish to ignore the precedent we see in other species, because these mutations occur across the board in everything. There are 3 different human piebald genes, 3 of them.
There is?
And there's 5 different types of albinism in humans. 5. We've got the same morphs as they do.
We have this piebald in humans, Nick?
Yeah. There's 3 different types of piebald in humans.
What does it look like?
It's usually, I mean, it can be as minor as... You know, somebody have like a skunk patch in their hair, and they got one gray pigmentless patch of hair in front of their head.
Yeah.
It can be as minor as that, or affect other parts of the body. But the fact that we have morphs of people too, we're not special. These things happen in everything. Yeah. You don't see them. You don't see human morphs very often because we are really opposed to inbreeding.
Yeah.
And so we culturally are so averse to that especially recessive alleles rarely pop up. Because, you know, it's selected against. Whereas it's also why you see so many albino snakes. Why the vast majority of snakes in the hobby come in albino. Yeah. And almost all those albinos are found in the wild because snakes don't have that hang-up about breeding with their relatives. And most snakes...
True.
And honestly, like a carpet python in the wild, the most likely mate that it will find is something it's fairly closely related to because they don't have very large ranges.
True. Very true.
So you see, well, there was 2 albino Darwins. There was the one that was found in 1992, Then there was one found about 12 years later in the exact same place. And for
Makes
a while
sense.
they tried to make it out like it was a new line. It's like it was in the same place. It's clearly not.
Same parents, probably. Right.
And it promptly got absorbed into the others. You don't hear about it. There were 2 leucistic spotted pythons found, 2 of them, in the same RV park, a decade apart, because clearly the local population around that RV park, where people go to pitch a tent, has a high frequency of hets that are all related, and occasionally they bump into each other.
There's a pond in Long Island that my friend told me about, and they see these musk turtles. They're finding like albinos in the pond. So this kid went and he captured 2 adult, regular-looking musk turtles. He brought them home. He bred them, and they may not be musk turtles. So I don't want to misquote, but it's something like that. And he bred them, and he produced albinos. So there's obviously an albino gene that's in that one pond. It's pretty big, but it's an isolated, genetically isolated population. So you get one albino, and everything's going to eventually get it. At least be a carrier of it.
Well, see, that's the thing. It's like being a visual albino is pretty detrimental to your survival, but being het for albino is not.
No.
So what you have is probably very few. Occasionally, you'll see a visual that hasn't gotten eaten by a bird yet.
Yeah.
But the hets are everywhere.
Right.
And it persists. There's Japanese rat snakes have a ton of wild albinos in one particular area.
Oh, interesting. Final question, Nick, because I don't want to keep you too long. If you had to pick one species that you don't currently work with, which would be a dream species for you, something that you don't have but you want to have in the future, what would you say it is at this point?
Oenpelli pythons.
And do you feel that you'll have them at some point within the next 5 years? Do you think it's possible?
Not in 5 years. I think before I die, I absolutely will. I think that the history of the hobby has shown that. What will happen, it'll probably be, they're now in private hands and multiple people have them in Australia and institutions have them. Eventually, every Australian zoo that wants them will have them. Then American zoos and European zoos will get them from those zoos, and eventually somebody's going to get some somewhere and sell them to the private sector, which is legally permissible under certain circumstances, usually in Europe. And then at that point, I will have them. I'm not holding my breath. I suspect it will take a good long while, but I'm patient.
What were the odds of Owen? What were the odds of Owen and Eric running into those in the wild? Was that like a 1 in 10 billion?
No, I think that like some people just have that sort of luck. Like really, like I've been there looking for him not once but twice.
Yeah.
I'm gonna be going back this year again, and I just like to get a picture of one. I'll be on my 3rd try, and of course they just go like, and I'm sure knowing Owen and Eric's luck, they probably just walked up like, oh look, there's an Oenpelli python.
Yeah, exactly.
It's probably just that easy.
It's Murphy's Law.
I know a guy, and he worked in the town of Oenpelli, and every night he's a teacher teaching at Aboriginal school there, and every night for like 2 years he walked that escarpment looking for those snakes. Every night for like 2 years. And then he found nothing for 2 years. Then he found 2 on back-to-back nights. He found one, then the next night found another one, and then nothing ever again.
Wow. Yeah.
Some people have that luck. I apparently am not one of them.
Yeah, well, I'll be interested to see when you finally get those. I think those will probably be the next big thing. You think they'll displace?
Yeah. I mean, I anticipate that will be a good long while, but it's got to go.
Yeah.
Right now, I don't think they've sent any to any foreign zoos, and it's usually that sort of a thing. And
yeah.
There are some countries where, like the United States, like a U.S. zoo, if they get an Australian species, an AZA zoo, they will not sell those animals. They won't hardly sell anything to the private sector. It's pretty difficult. But there are also private non-AZA affiliated zoological facilities that can get stuff in legal zoo transfers on occasion if they're willing to jump through a lot of hoops and paperwork. And they're not bound by the AZA rules and whatnot. So eventually it will happen.
Yeah. Do you have Boland's pythons, Nick?
What?
Do you have Boland's pythons?
Nah, I don't really want them. I can't get, well, it's not saying I don't want them. I know what it would require to be successful with them, and
Yeah.
I can't realistically provide that, so I
Gotcha.
do not keep them.
Yeah.
I don't have, I mean, what Bolin's pythons need is what people are very reluctant to give them. They cannot get, they cost a lot of money, and we have become conditioned as a hobby to use food intake and feeding response as a barometer of overall health. Because a healthy snake will eat, and a sick snake won't eat.
Right.
So when you spend $9,000 on this pair of Bolin's pythons, you really want to see them eat, because when they eat, you think, oh, I must be doing it right. It's eating, it's healthy.
Right.
And they get way too big, way too fat, and die way too young and never breed.
Right.
And no one ever figures this out. It's like the reality, these snakes should be, no one wants to believe, you know what the secret to breeding Bolin's pythons is? Keep them about 7.5 feet long, skinny as a rail with a big ass head. They should look like a 7.5 foot Amazon tree boa. But nobody can bring themselves to spend $9,000 on their dream pair of snakes and then proceed to freeze their balls off and starve them half the time. They can't do it. Yet that's the secret to success. I mean, Mark Spataro is an old friend of mine, and he's been there a number of times and even found an adult female brooding a clutch of eggs in the wild.
Yeah.
I said, Mark, in all of your time finding all these ones in the wild, he goes, what was the biggest one you ever saw? He's about 7-8 feet and skinny.
Wow.
I'm like, then why the hell is this snake 10 feet long? It's like, why is this so big? Like, it's like they're not supposed, just because something can grow to a given size doesn't mean that it should. And people are constantly conflating two separate things.
Yeah.
And those are maximum size and adult size. And those are very different.
Sure.
Like, if you were to ask me, like, you don't judge the average height of the average citizen of the People's Republic of China by the height of Yao Ming. He's one Chinese dude who's 7 foot and over 7 feet tall. You would say, well, I guess by that logic, I guess people in China get 7 feet tall. That's insane.
Right.
Everybody's 2 foot shorter than that.
Sure.
Like, anacondas get 32 feet long. It's like, no, the average adult green anaconda female is 17 feet long, not 32 feet. You can't judge by the outliers.
Of course.
You can't, it's like, and that's what people tend to do. So while, yes, your Bolin's python could conceivably get 12 or 13 feet long, a good reproductive size, 7-8 feet is probably right in the wheelhouse for success.
Interesting.
And what you find is when things get to be very large, their reproductive fecundity and fertility go in the toilet.
Interesting. That seems to be the theme of the show. I know Vin Russo's a big believer of that with boas too. You just got to stop feeding them so much, keep them smaller, they'll live longer and they'll breed better.
Well, a lot of people think that snakes are always supposed to be round in cross-section, but you know this, you've got a lot of boas.
Yeah.
Boas should look like a loaf of bread, flat in the bottom, flat in the sides, and rounded on the top like a loaf of Wonder Bread.
Yep.
Like, slightly laterally compressed. That's a healthy, lean, muscular, lean snake.
Yeah.
And that's what they're supposed to be. When you try to make them round, that's why people have terrible luck with womas. Womas are supposed to be shaped like a freaking Hershey's Kiss. So are blood pythons. Round the bottom, taper the side, a triangle with a ridge down their back.
Right.
And people think they look emaciated when that's what they're supposed to be, how they're supposed to be built. And they trying to make them round, and they make them morbidly obese, and they don't do well.
Interesting. Well, Nick, I want to really thank you. I think this was a great educational piece we did. It was entertaining, and you're very knowledgeable. When's the new book coming out?
We don't know. I mean, the goal, I hate to even say it on camera because then everybody will try to hold me to it. The goal is late this year, but it requires a bunch of lead time to get it all formatted and then printed and shipped and all of that. So the goal is very much to have it in people's hands this year.
Yeah, but by the time you get done with it, it'll be 1,000 pages. That's the problem. The focus is changing a bit now. There's give me far fewer captive animals in the book and a much greater focus in the species chapters on wild snakes. Sure. Because I love nothing more than seeing like beautiful images of wild snakes. Yeah, it's great. In the wild. I've got some. I just got some from a guy today. Some inland carpets, and these are just stunning pictures in the wild, untouched, just as they walked upon. Oh, look, there it is, and like with the habitat and the contact. So there'll be a lot more of that. The captive sections about breeding and husbandry and all of that will have ample pictures of captive, and selectively bred animals. The morph chapter is one of the things one of my responsibilities, and it's getting almost unmanageable in its size and scale now. I can imagine.
My guess is it might end up being 80 pages.
Wow.
It could almost be a book unto itself.
I was going to say, yeah, definitely.
And if I say 80 pages, that's if I only give, each base morph 2 pictures and each combo a single picture, 2 or 3 to a page. It will take that many pages.
Interesting. How much do you think the book's going to cost when it's all said and done?
I don't know. That's up to Bob. But there'll be a much greater explanation of how morphs work, how the pigment distribution versus synthesis,
right? How
they work at the molecular level,
sure,
and inheritance, and a lot of the more academic side of it. There'll be a much greater. I'm writing a section on how you prove out a new morph, because I'm sure you've seen it a bunch of times. A bunch of times where people they will have something they think they'll jump the gun and think they declare they've got some new mutation that they have not done their due diligence and properly proven out, and so there'll be a section on how you go about doing that.
That's definitely something that needs to be done, because I think people. You're right. Everyone thinks they got their own new morph in their collection.
They always jump to the conclusion that is most beneficial to them.
Exactly.
And it's like what they need to be doing is a series of very precise, controlled test breedings to get definitive results and eliminate other hypotheses.
That takes too long, Nick. Come on. No one's going to do that.
It took me 5 years to prove out those stonewashed metal pythons. 5 years.
Nick,
I've got other things I'm trying to prove out now that have been years in the cooker before it's time to. It's just how you're supposed to. But there's the right way to do something, and there's everything else that's not the right way.
Yeah, exactly. Well, your website is inlandreptile.com, and you got 1 million animals up there listed. I saw recently. If you can't find something carpet-wise on that website, then you don't know what you're looking for. That's for sure, because you got some great stuff on there. I've bought animals from you. Every single animal I've ever gotten from you is still alive, very healthy, and you produce some great stuff. I want to just thank you for really, putting. I mean, it's amazing how many animals you produce, and every one of them, you stand by, and they're all top quality.
Thank you very much.
You could send me my commission check tomorrow.
There you go.
No, I'm just kidding. No, he really, Nick is super professional, and of all the people I've dealt with, probably one of the best guys in terms of if you want to buy a snake, and he's very, very helpful in terms of information and everything like that. And that to me is important. When I first got into carpets, I didn't know that much about it, so you always answered all my questions, and every time I bought a snake from you, you never hesitated or act like I was irritating you or bothering you. And not all breeders do that, so that's I have to commend you for that.
Well, thanks.
All right, guys, but that's going to take us to the end of another episode of Muscle Serpents University. If you guys like what you're seeing and you want to see more of Nick, if you had any topics, let us know. I'd love to get him back again. For now that we are out of time, guys, if you like this channel, make sure you hit that subscribe button, turn on your notifications, hit the like button, and guess what? We will see you back next week.